Good morning. My name is Casaccia, and thank you for inviting me for this research day on Alzheimer's disease and related disorders. I am from Italy, from this small region here with the letter called Abruzzo. And that's where I went to high school. And this provided me with the opportunity to learn English. My parents did not speak anything else than Italian, but I was able through scholarship to go to London and learn English. For medical school, in Italy, we can go to medical school right after high school. So I went to medical school at 17 in Rome, and that's where I became very interested right away on the brain. And then I did my internship in an electrophysiology lab on sleep and evoked potentials. And it was while I was an intern that I was able to diagnose multiple sclerosis to my aunt first, and then to a friend of mine. At that time, there were no therapies for multiple sclerosis. And so I started the residency in neurology, but I really could not stand it. It was really difficult for me to be a neurologist and not having any therapy at hand. So I discussed it a lot with my director, and that's how I decided to come to New York. And I came to New York with a scholarship from FIDIA to study prions and prion diseases in prion diseases. But as soon as I came, I realized that as an MD, I really wanted to understand the mechanism. And so I left the residency in Italy and I started a PhD here on molecular and cellular neurobiology. And ever since, I've been studying myelin and oligodendrocytes. And more recently then, I've been starting to studying also mechanism of neurodegeneration. But pretty much this is multiple sclerosis and the level of therapies. The asterisks indicates when I came to the United States, very long time ago. And what you can see is that when I came, there were no therapies. And then since then, there have been a number of new treatments that have been developed. But something that still remains a major issue in multiple sclerosis is the fact that while several patients have a course that it is relapsing when they get some kind of symptoms and remitting, patients feels better, everybody, regardless of the level of inflammation that they might have, they tend with age to go into a progressive stage where there is a critical clinical disability, which is characterized by high level of neurodegeneration. And so I've been really interested coming from this world of multiple sclerosis to understanding mechanism of neurodegeneration. And more specifically, I was interested in the fact that neurodegeneration was something, and the changes that we could see in MS were shared with several other neurodegenerative disorders. In terms of mechanism related to neurodegeneration that are neuronal intrinsic, an interesting finding was the fact that nuclear export was critically affected in the neurons from multiple sclerosis patients. And this led us to identify potential therapies to alter this nuclear transport and then experiment those therapies in animal models of multiple sclerosis. In addition, another important mechanism besides altered nuclear transport is mitochondrial degeneration, mitochondrial dysfunction in those cells. And this has been the work that also Maureen and Achilles and Hygiene have done in the lab, where we start studying the cerebral spinal fluid and the plasma of patients with this progressive disease and neurodegeneration. And on a separate level, we have had additional studies characterizing the blood of multiple sclerosis patients. So altogether, the approaches that we have tried to use range from going from patients, from studying samples from patients using different omics, to testing hypotheses in mice, to try to identify therapies in humans. So just to recapitulate, I think that in terms of overall interest, we are interested in addressing this devastating societal problem, which is neurodegeneration. We all know they are on the rise, and we all know that no effective therapy is currently available. So our current approach is from a standpoint of metabolism. What is the role that metabolism plays in neurodegeneration? And can we use, in our case, nanoscience or bioengineering to design targeted sequestration approaches? And this was an interesting studies that we had from a student in the lab, Camila Castro, that was recently published in eBiomedicine, where the approach, as mentioned before, was to just take the clinical disability in the patients, follow the MRI, and then performing this omic analysis, lipidomic, metabolomic, and epigenomic. And more recently, we tried to repeat a similar study in healthy control versus MS patients, or in MS patient that we followed longitudinally. And what we were able to detect was the presence of specific metabolite, this time not only in the blood, but also in the cerebrospinal fluid of patients. And we were able to distinguish metabolites present in patients that remain relatively stable over a period of time from those in patients that decline dramatically within a 2-year time period. And something that was to us extremely exciting is the fact that many of the differential metabolites are actually produced by gut bacteria, thereby highlighting the importance of gut-brain communication. And this is the type of work we are interested in continuing to perform now, apart from this metabolite, and work with chemical engineers and nanoscientists in order to test potential mechanism of regulation of metabolites in culture to eventually lead to potential edible therapies. Thank you.